Next speaker is Amir Horrowitz. Amir studies a cell type called the NK, or natural killer cell, that we have learned in the last, I would say, 5 years is an important component of our immune response to tumors, and I think the data is even becoming more and more convincing.
All right, well, thank you very much, Steve, for the introduction, and thank you so much to offer me the opportunity to present my group's focus. So my group focuses on human natural killer cells, or NK cells, and the immunogenetics that underlie or govern their capacity to respond to their environment. So shown up here is highlighting our group's central hypothesis, which is that expression of HLA class 1, and specifically the differential expression of HLA A, B, and C, as well as the non-classical and conserved HLA-E on a tumor cell, actually has a profound ability to determine an NK cell's capacity to respond to this tumor. So shown in the schematic here, you have in settings where a tumor cell has very high levels of HLA-E expression, and this elicits very strong inhibition of NK cells. This is presumably happening at the very earliest time points following tumorigenesis, at a time point preceding when T cells have seen their antigen and had the ability to proliferate to sufficient numbers. And so this associates with tumor escape. Now, when we look at a different situation where a host's genetics promotes low expression of HLA-E, this is what we find on the tumor cell, and you have a lower threshold for NK cell activation. And so what we see here is the ability of NK cells to directly target the tumor cells, causing lysis through perforin and granzyme-mediated approaches. But also very critically, NK cells produce very rapid and abundant supplies of chemokines and cytokines, which are critical for potentiating this very well-established crosstalk between antigen-presenting cells, T cells, specifically CD4s and CD8s. So most recently, my group and several collaborators have been characterizing HLA class 1 expression, and what we find is that with respect to what regulates HLA-E on a cell is HLA-A and HLA-B and HLA-C. They provide a monomer peptide that's very conserved, and uniquely to A and B, which are considered the dominant regulators of E expression, is we find that HLA-A expression is extremely variable when you look across the alleles. However, HLA-B, which, just to set the context, is the single most polymorphic gene of all the HLAs, let alone in our genome, it's actually quite uniformly expressed. And so what's really important to consider here is that every single HLA-A allele encodes a good HLA-E binding peptide. However, approximately 80% of all HLA-B alleles encode an unsuitable peptide. This leads to unstable expression of HLA-E, which associates with very low cell surface expression. So just highlighting an example from a recent study of ours that was published in Science earlier this year in settings of HIV infection, is that the way that HLA-A and HLA-B are associating with natural immunity to viral infection is really quite profound, as shown by those curves. So in a very small pilot study of 60 healthy people, we actually empirically measured HLA-E expression on peripheral blood mononuclear cells, and we find that there's a strong correlation, albeit, well, rather a highly significant correlation, albeit not terribly strong. However, when you parse out these donors such that you can actually see whether the B alleles encode a good or a bad peptide, and then you can qualify along this spectrum of how HLA-A is expressed, you actually see these profound correlations. So it's very exciting. Just highlighting an example of NK cell activation, really to highlight the point that when you look at activated NK cells, they're actually mirroring the direction of their education. And so we can look at this dynamic range here. In blue are individuals that have B alleles that encode good E-binding peptides, and in yellow are individuals homozygous for B alleles with bad peptides. And you can see here that in individuals with higher HLA-E, The direction of the NK cell response is actually very closely related as well to how A expression is found on these cells, and especially as well the NKG2A expressing NK cells that interact with HLA-E, they are the dominantly activated subset. We see the exact inverse association in individuals with low HLA-E. So just to summarize, my group focuses on immunogenetics of HLA class 1 genes, KIR genes, NKG2A and C genes in cancer patients receiving immunotherapies. We're specifically using whole exome sequence data to compare germline and tumor tissue. We're also very much focused on ex vivo profiling for antigen presentation pathways associated with HLA class 1 expression, but also NK cells and T cells, as well as myeloid cells within the tumor microenvironment. And I won't go into the technologies, you'll hear them in a later session, but these are very big focus of what the Human Immune Monitoring Center here at Sinai offers. And in doing so, we're also studying mechanisms that actually restrict infiltration of NK cells as well as T cells into the tumor microenvironment. In settings where you're actually losing HLA, you're actually seeing certain soluble signals suppress it. And finally, studying abrogating inhibitory pathways as combination strategies. So with that, I collaborate very closely with a number of people here at Sinai. My group is small but growing, and thank you very much.